Hello, welcome to the Herbarium. You're coming here live from the Herbarium, the Lower Herbarium, in fact, of the Carnegie Museum of Natural History in Pittsburgh, PA. My name is Mason Heberling, and I am a postdoctoral researcher here in botany. So, you might ask, so this is behind the scenes. The normal visitor doesn't get to see this. You might ask, what is the herbarium? What is an herbarium? You might also already know what an herbarium is, but I'll tell you anyway. So an herbarium is one of the many collections, one of the what we call them biological collections, natural history collections, here at the museum. And in particular, it's a collection of plants, most of which are dried, pressed, dead plants to a sheet. And so this is an herbarium. The plural of an herbarium is herbaria, just to make it confusing for you, but I might say herbaria here and then, and that's also an herbarium. And today I'm going to talk about the digitization project that we're working on. So this is just a typical herbarium specimen, a flat, dried, dead plant, glued or fixed to an archival sheet. This one in particular is a wildflower that I love, Trillium grandiflorum, the large flower trillium. And here this was collected near Old Morningside Road, 18th Ward, Pittsburgh, May 4th, 1884. And so, ta-da, this is an herbarium specimen. And so we've got a lot of really old specimens, but I also want to make sure that it's clear that we got a lot of new specimens too. So here also is a Trillium grandiflorum, the large flower trillium. And this was collected by Bonnie Isaac, the collections manager here, May 5th of this year, 2018. And so not a ton has changed in the past 150 plus years of the collections here. But there are a couple of important information here, including what the specimen is, who collected it, when it was collected, where it was collected. So in this newer specimen, we have latitude and longitude, so put it on a map, we know exactly where Bonnie collected this specimen. Whereas this super old, equally beautiful specimen does not have latitude and longitude, as you might guess. John Schaefer, one of the first curators here at the museum, did not have his GPS with him when he collected this. I'm making a joke because that technology didn't exist in 1884. It's a tough room here with the live thing. It's hard to tell if it's funny, but I assume that there's immense laughter on the other end here. So there's a lot to the specimen, both the specimen itself, so the physical material, this plant was growing up until it was picked in 1884. And then almost equally as important, if not more important, is the actual label information. So we know where it was collected and when it was collected. And so this is the Carnegie Museum herbarium, and there's many herbarium all across the world. So if we could put up a map of the different herbaria of the world. So there's an estimated more than 380 million specimens that exist worldwide in over 3,000 herbaria. Herbaria is the plural of herbarium. And the Carnegie Museum is one of those collections, and it's a very important collection, and as you might guess, it documents the plants that exist now and have existed in the past, particularly in western Pennsylvania, but the collections are also worldwide. So here in this herbarium, it's actually split between two rooms. There's more than 530,000 plant specimens that take the form looking similar to this. So think about 530,000 of these, so over 500,000 of these, collected through time. And so you might ask, so now you know what an herbarium is, you might ask, why do we have these specimens? And so that is a valid question. And there are a lot of different purposes. So a main purpose of a specimen and having these specimens are to document what lived where and when in a verifiable way. So John Schaefer could have just said, hey, I saw some trillium there, and you'd be like, oh, okay. And then 50 years later, we may or may not know that he actually saw trillium there. And so we know that trillium was, that this plant species was there. Another big reason is for taxonomy, so studying the tree of life, so understanding how plant species have evolved and or are related to each other. So for floristics, which is the study of what grows where in a particular region. Species identification, so if somebody finds a plant that they don't know, they have a reference collection to look at and be like, yes, this is Trillium. Or they might discover a new species, and that a lot of species discovery happens here in the herbarium. Another is voucher, what we call a scientific voucher. So I said the word verifiability, so the scientific method is all about repeatability and verifiability. So if Sarah over here says that she saw something, we could believe her, but if she actually had the raw data to show us, which would be the specimen, then we'll definitely believe her, and 100 years down the road, when she published a paper on that, we'll know exactly what plant she was talking about, because our understanding of what we call that plant may have changed in 100 years. And then, of course, education, which I like to think is what's happening right now, hopefully, but also just being able to use this amazing collection to, you can visit all seasons of Pennsylvania right here. You can visit other states right here, and you can visit across the world and learn about the flora or the different plants that exist across the world. And then there's much, much more. And so today I mostly wanted to talk about an active, ongoing project funded by the National Science Foundation, a digitization project. So museum digitization is a huge thing in museums now, and that is essentially turning these specimens, these physical specimens with label information, putting it in a digital format, and we'll talk about why we want to do that. And so the Carnegie Museum, again, funded by the National Science Foundation, we've joined the Mid-Atlantic Megalopolis project. I think I got a map there to show you. It is a bunch of herbaria, multiple herbarium, in the Mid-Atlantic region, where we're digitizing specimens across the region of Pennsylvania, New York, Washington, D.C., Maryland, Delaware, and New Jersey. And so we're joining this digitization network, which is led up by Dr. Cindy Schema at the Morris Arboretum in Philadelphia. And so as you see from the map, Pittsburgh is kind of a densely populated area, along with the dense, what we're calling the Mid-Atlantic Megalopolis from Washington, D.C. to New York City. And so Pittsburgh has an important industrial past and an important kind of hub for the transport of human activity across the Mid-Atlantic and beyond. And so over the next 3 years, these people you see behind me, these wonderful people, will be working to digitize about 190,000 of our 530,000 specimens. So I said we have 530,000 specimens, about a third are from Pennsylvania, about a third are from outside Pennsylvania, and about a third are all across the world. So we are just digitizing to start, digitizing the specimens in the Mid-Atlantic region. And so once we have these data in a kind of a searchable form and in a way that we can look at images, for instance, we can see how the flora or how the plants have changed in the context of urbanization. So as cities and human populations, and we've started to do things to the environment, we can see these effects. So, without further ado, I wanted to just point to what the heck these people behind me are doing. These people. And so Sarah Williams here is the project manager of this Mid-Atlantic Megalopolis, our input of the digitization happening here. And she's working here today with 2 work-study students from Pitt, Evan Moran, I'm going to get the names right and pronounced right. This is live after all. And Stephanie Iannotti. All right, I did it. I would hate to say her name wrong on the live to the millions of viewers right now. So what they're doing now is, you see this camera and kind of this fancy light box setup, is that they're actually working to digitize our grape specimens in the genus Vitis. And so all of the label information has already been online, largely thanks to the efforts of Bonnie Isaac, who I mentioned, who collected this plant specimen. But we don't have many images. But now we do have many images. How many images do we have up? We just started in September.
As of yesterday, we have 17,700 images online.
17,000 images. So right now, did I show the map 3 of the so this should be a map color coding where the specimens are that this crew is imaging. So you can see a lot around Pittsburgh. And so that's why our collection is really important, because we have a really, really strong collection that dates back to the mid-1800s to see the changes in the plants and the plant communities through time. So this is kind of the workflow. It might seem, I don't know if you can really see much, but they're essentially taking high-resolution pictures of these 190,000 specimens. They're doing what you see right now 190,000 times, plus some other digital manipulations to get it online so it's freely accessible. And so you might ask, why would we want to have these imaged? And it really is all about accessibility. So even 10 years ago, if someone wanted, if either an interested student or a scientist, even halfway across Pennsylvania, let alone halfway across the world, wanted to see some of these specimens, they'd either need to request a very specific loan. They'd be like, hey, I want to see this trillium from 1884. Or they would need to come and visit the facility. And now, all of this information is online. And so I wanted to go to another image here, more of a seasonal image. You could go to midatlanticherbaria.org, and that's where this team is putting up these images. Let's see here, yes. And so here I think I just did a little screenshot of searching red maple, or the scientific name Acer rubrum. And then you could go to Mid-Atlantic Herbaria, search that. You could search to see what was collected on your birthday, see what was collected around your community, see what was collected in Asia, see a particular species. Here I just did a search of Acer rubrum because it's a beautiful fall tree that has some beautiful foliage. And if you go to the next, my next little still shot is you can even map. So you can see where red maple, where our collection is, and where it has been collected, both through time, so since 1850 onward, and through space, so across Pennsylvania and across the Mid-Atlantic region. So this is just the Carnegie Museum's input of this Mid-Atlantic Megalopolis project, but with all the other herbaria, you really can see which plants existed when and where, and what has changed through time, and you can do these really cool maps, and we call that Geo Referencing. So if it didn't have a latitude, longitude, to put it on a map, put it exactly on a map. That's also what this team is working on, is to put, for some of these old specimens, to be able to put it on a map, and that also opens up a whole new line of research that would otherwise not be possible. And so I just pulled this one because I think it's beautiful, but this is, I think, what was on the still shot too. This is red maple. Collected on Chestnut Ridge, vicinity Hillside in Westmoreland County, which is just outside of Pittsburgh, by an early influential curator here, Otto Jennings, in 1922. So 96 years later, it's still got this rockin' foliage. I'm allowed to say rockin', right? Yeah, okay. So then, in addition to having the images, so experts across the world can look at can use these data and to do, even to identify specimens that we may not know or may have been misidentified. Experts now can just sit at their desk and see all of our collection. So that also improves the collection. So it improves research outside of the museum, it improves research in the museum, and it also just makes our collections available to the public. So why does it matter? We can access it virtually. So I'm an invasion ecologist. I study introduced plant species, and for some of my main research, I do use images looking at, for instance, the introduction of an invasive weed when it was introduced into Pittsburgh, into the area, what it looked like. So if the leaves were big or small, for instance, if it was tall or short. And then you can also look at that through time because it's been collected repeatedly in Pittsburgh, for instance, a particular weed. So that is one use, and so if you want to show another, my next image of doing some digital analysis of leaf morphology, for instance, or leaf shape. So with an image and computer technology, we can now measure kind of the fine details that would otherwise be either really difficult or impossible. So that's that, and so I didn't want to like steal the show. I don't know if you guys wanted to say anything in particular. So I think Sarah had a couple stats of where we are with the on the Mid-Atlantic. How many families have we done so far? How many? You said we have 17,000 images online. We just started.
17,000. There's 280 families that we have our herbarium split up into, and so we've made it through 42 of those just through Pennsylvania. We still have the other Mid-Atlantic states to go, as well as some cultivated plants and some undetermined plants thus far. So hopefully once we get those imaged, we may even get some help in identifying what they might be.
Right, so we have a lot of specimens that aren't determined to species. So now that they're online, experts and other people can look at them, and maybe they know what they are, and so it kind of opens up the cabinets to a whole new community in a new way. So yay. Okay, so I think we got some questions. I got a ton more specimens. I could talk all day about specimens, but in fact you could look at all these specimens yourself too. All right. That's a great question. So you might want to know what the question was, huh? Maybe I should do a Jeopardy style answer it and then you can guess what the question was. No. Have you discovered or found out anything new during digitization? So that's a great question. Maybe that would be a good question for Sarah. I'll give my take on it first. That this is super important, and so just the act of digitizing means that they're going through 190,000 specimens and they're able to look at them. And some of these specimens may or may not have been touched in a while, or they may or may not have. I don't want to say they haven't been touched because there's a lot of active research going in with each of these specimens. But kind of looking at it from a new perspective and maybe pulling out one specimen that looks like an outlier in a folder, those sorts of things. So have I personally discovered anything new during digitization? Man, live TV, I have to, I don't know if this is TV, Facebook, anyway. Sarah, have you discovered anything?
I know that we all kind of discovered a really cool plant that we all liked. We were imaging the Rurus typhina, which you may know as Staghorn Sumac, and it has these really great furry twigs and furry bright red fruits. And you know, I know for me, certainly after seeing 100 or 200 of them while imaging, I will be able to pick that out whenever I'm out in nature. And
yeah.
I don't know about you guys, I know we all thought that they were adorable.
Yeah, so it's a good learning experience for everyone involved.
Yeah, do you think you'd be able to find that plant and know what it was?
So many different kinds of them. It was amazing.
So yes. That being said, a lot of other herbaria, they don't even have their label information online. So I said Bonnie Isaac already they're already online, which is a huge endeavor and a huge part of digitization. So the whole collection is searchable now. There's a lot of images coming on, but it's entirely searchable now. But for other institutions that they don't even really know what they even have in their collection, because they're also, we call transcribing the label information. So taking their handwritten labels and putting them in a searchable database. So I'd imagine those herbaria in particular are like, holy heck, I didn't realize that we had that. So, all right, another question. Do you still need the specimens once they are digitized? That's a great question. So digitization is super important, and I also don't want that to come across that the original, once we take a picture of this beautiful Acer rubrum, for instance, we don't need it anymore because we have all the information we need from it. That's not true. So there's a lot of information that is on this sheet that cannot be seen from an image. One of which being, so you can zoom in, I meant to mention, you can really zoom in these high resolution photos and see a lot. But you can only zoom in so far, I'd say, and of course there's nano, small fine scale detail on here. It's hard to get DNA from an image. I challenge someone to do that, it might be pretty difficult, but having the actual specimen, you can get DNA from an image. So yes, you definitely still need the specimen once they're digitized, and in fact, being able to, having this digitization project happen, it really does make the physical specimens more important, in fact, because now that it's out there on the net, there'll be experts that want to come in and see the specimen, and they may otherwise have not known that we had that particular specimen, for instance. All right, so With that, I think I've probably talked a lot and enough. Okay. Go to midatlanticherbaria.org, check out what we have. I encourage you to use the images in any way, if you want to print them out and just look at them, or if you want to use them in education, or just scroll through, see what was collected in your neighborhood. I'd like to thank Sarah, Sarah Williams, the program manager of the activities happen here. So keep an eye on that. And of course the National Science Foundation, which is, has made this research and activity possible. And with that, thank you. I think we wave now, yeah.
Bye.
Evan, wave.